Okay, irreducible complexity. Has it been rebutted to the point where an intelligent designer is ruled out? We go to index B. We go to the study. Bacteria flagellum refuted by evolutionists and then rebutted by creationists. Okay, and then we'll look up where we left off. Secretory apparatus. The type 3 secretory apparatus. We're looking at bacteria flagellum that was used, its function was for locomotion, not to secrete toxins within the host by the bacteria to actually kill the host. Digest it, I guess. So in the popular imagination, Professor Miller says, bacteria are germs, tiny microscopic bugs that make us sick. Microbiologists smile at that generalization, knowing that most bacteria are perfectly benign and many are beneficial. I don't know what this point is. Some scientists are intelligent designers, advocates, and some are not. Some people don't know about germs, some do, even essential to human life. Well, I don't know, most people do know that there's some benign bacteria and some ones that are toxic. I don't know what the point is. Nonetheless, there are indeed bacteria that produce diseases ranging from the mildly unpleasant to the truly dangerous. Pathogenic or disease-causing bacteria threaten the organisms they infect in a variety of ways, one of which is to produce poisons and inject them directly into the cells of the body. That's this secretory apparatus he's talking about, which is not the point. Can you produce this with fewer components that allow the bacteria, the particular species of bacteria involved in view, to locomote, to move around, not to secrete toxins? Once inside, these toxins break down and destroy the host cells, producing illness, tissue damage, and sometimes even death. That's interesting, but it's not apples and oranges. It's apples and oranges. In order to carry out this diabolical work, bacteria must not only produce the protein toxins that bring about the demise of their hosts, but they must efficiently inject them across the cell membranes and into the cells of their hosts. They do this by any means of any number of specialized protein and secretory systems. One known as the type 3 secretory system, TTSS, allows gram-negative bacteria to translocate proteins directly into the cytoplasm of a host cell. The proteins transferred through the TTSS include a variety of truly dangerous molecules, some of which are known as virulence factors and are directly responsible for the pathogenic activity of some of the most deadly bacteria in existence. Fine. What about locomotion? That's the subject. At first glance, the existence of the TTSS, a nasty little device that allows bacteria to inject these toxins through the cell membranes of its unsuspecting hosts, and seem to have little to do with the flagellum. So what? However, molecular studies of the proteins of the TTSS have revealed a surprising fact. The proteins of the TTSS are directly homologous or same to the proteins in the basal portion of the bacteria flagellum. Well, they have similar components in different species. So figure 2 below shows these homologies, okay, extend to a cluster of closely associated proteins found in both of these molecular machines. On the basis of these homologies, McNabb has argued that the flagellum itself should be regarded as a type 3 secretory system. Why? The flagellum in the other bacteria is for locomotion. That's the function. Why do you regard it as something that would secrete poison? It doesn't in that particular bacteria, but in others it does, but with less components. It's apples and oranges. Extending such studies with a detailed comparison of the proteins associated with both systems, this fellow has seconded this suggestion, noting that the two systems contain homologous component proteins with common physicochemical properties. The argument is psycho. It is now clear, therefore, that a smaller subset of the full complement of proteins in the flagellum makes up a functional transmembrane portion of the TTS. Well, they're similar, but not the same. So figure 2, that's fine. It's interesting. There are extensive homologies between type 3 secretory proteins and proteins involved in export in the basal region of the bacteria flagellum. These homologies demonstrate that the bacterial flagellum is not irreducibly complex. How do you determine that? You can use fewer components in another species to secrete poison, or use more components in yet another species to locomote it around. They do not demonstrate that the bacteria flagellum is not irreducibly complex, because there are 2 species involved in your investigation, wherein you are presupposing one system in one species is capable of operating with a second species, replacing a more complex system, and function perfectly well within that species for the function that the more complex system was performing. So you take away things, you say, well, if you take away a few items, it'll still locomote. Well, did you do that? No, you didn't. Or you offer a different function which would obfuscate the first function, which was locomotion, which would cause the rapid demise of the species. You didn't do that. And it appears that you have not tested this hypothesis. Just because you might observe a simpler transmission in a functioning Ford with components that are the same as in a Toyota, which has a more complex transmission with more components, this does not lead to the conclusion that the simpler transmission would work as well in a Toyota unless you properly test this hypothesis. The Ford might be similar in design relative to a number of components identical to those in a Toyota, but you do not have sufficient evidence that the simpler transmission in the Ford could perform the same or another kind of function in the other Toyota car, which is more complex, and it's a Toyota, such as a carburetor. This point was never thoroughly tested. Besides that, another function would leave the Toyota without a transmission, leading to its rapid demise. No control, no falsification, no repeatability. The overall complexity of the Ford versus the Toyota and how all of its components function together in performing all the function was not investigated. After all, Ford is one species and Toyota is another. So the evidence of whether or not a life form was created by an intelligent designer or evolved from another or other life forms is scientifically dependent by and large upon that original life form, not upon another species from which one is guessing it evolved into and from, which has a different history of existence, and especially not if the species has significant differences from the original species, which most species do. Cherry-picking features without presenting detailed evidence from one species without connecting the dots of evidence to another species is hardly scientific. 2 or more species that are of similar design may well have been created by an intelligent designer besides, with significant yet original differences as opposed to evolving from a common ancestor in a non-identical manner. The point is that if either species evidences that all components must be available and in place at the same time in order to function properly, then that species is irreducibly complex. Probability is statistically wise not evolvable, because it would be going beyond the age of the universe. It must have been created by an intelligent designer, one species at a time then. Just because you can prove your case doesn't mean you've then disproved an intelligent designer. In this diagram, which we looked at, the shaded portions of the basal region indicate proteins in the E. coli flagellum homologous to the type 3 secretory structure of Yersinia. So hard to pronounce. And we looked at that. Stated directly, the TTSS does its dirty work using a handful of proteins for the base of the flagellum. From the evolutionary point of view, this relationship is hardly surprising. Yes, it is. It doesn't mean that it's not going to function differently in another, and if it's put together differently in another species. In fact, we have components in the human body that are similar to components in a dog. But that doesn't mean that you can take the components in the dog, which might be simpler, and take them out and put them back in the human and it'll function. In fact, it's to be expected that the opportunism of evolutionary processes would mix and match proteins to produce new and novel functions. We're not saying it doesn't, produce the components, but we're not saying they evolved. Have you taken out parts? Have you put it in part and then added more parts to see if it continues to function or advance? So the evolutionary point of view is largely one of innumerable repeated failures over eons of time until something of value is produced with probability-wise, is usually older than the universe is estimated to be, hence unlikely, hence not plausible. Hence the more plausible conclusion is that the species was created by an intelligent designer instantly and recently. Even if plausibility is significant for the evolutionary model, this does not rule out the intelligent designer model, which is in any case far more convenient, efficient, and promising, and quicker. For the evolutionary mindset is hardly an opportunistic process because it is built into their thinking that there will be innumerable failures until one minuscule success is achieved, and then billions of years later, we got something working. Furthermore, the TTSS species is not the same as the flagellum of a bacteria, nor the same as the species of the other bacteria that has that flagellum for locomotion. There are similarities amongst these two species, but they are not identical, especially not relative to the question as to whether either or both species was created by an intelligent designer or evolved over eons after the necessary innumerable repeated failures. According to the doctrine of irreducible complexity, however, this should not be possible. Well, within the same species, so far, it has not proved to be possible to put the simpler component referred to the TTSS into the more complex bacteria and have it function properly. That wasn't done, either as a replacement method of locomotion or as a dispenser of toxins. If the flagellum, this guy says, is indeed irreducibly complex, then removing just one part, let alone 10 or 15, should render what remains by definition functional, non-functional. Has this actually been tested? No. You don't stipulate that you've done this. So how can you conclude that the species in view does not need the specified components and yet still operate as before? So take out some components in the locomotion bacterium from their flagellum and see if it continues to locomote. That's basic thought there, logic. Yet the TTSS is indeedly fully functional, even though it is missing most of the parts of the flagellum, not as the locomotion in the original species, only as a toxin secretory mechanism in the other species. The TTSS may be bad news for us, but for the bacteria that possess it, it is truly a valuable biochemical machine. Yes, but was it evolved or was it created by an intelligent designer? You didn't try that either. The TTSS and the flagellum of a bacteria are not the same species, apples and oranges. If the TTSS is a legitimate species to consider, your examination of it was insufficient to prove that the flagellum of a bacteria could indeed operate properly with the same system that is in the TTSS as before, or operate properly but differently. You haven't provided that proof. The system in the TTSS is different from the system in the flagellum, so it is most likely would not operate in the same way to provide locomotion for the bacteria or any other way, because you have not stipulated that you have found such bacteria in existence which does. You got to find an example of that, or try to manipulate it yourself. The existence of the TTSS in a wide variety of bacteria demonstrates that a small portion of the irreducibly complex flagellum can indeed carry out an important biological function, but not the same. But not the same function as in the different species. Since such a function is clearly favorable by natural selection, the contention that the flagellum must be fully assembled before any of its component parts can be useful is obviously incorrect. You haven't proved that. Science is faulty. Have you placed the mechanism in the TTSS in the bacteria to test this that has the locomotion? What this means is that the argument for intelligent design of the flagellum has failed. Wow. So you're testing the wrong hypothesis, proves something that is irrelevant, and boasted that you have defeated the concept of intelligent designer. But even what you have determined is true, but even if that is true, and it is not, an intelligent designer could have created both species from nothing in far less time, and you haven't checked all the species out there to see that something is not qualified through evolution to be arrived at. There's got to be something out there, we don't know it yet, that's more intelligent than evolution can handle. I can use your argument too. We don't know yet, but we'll find out.